interactions on Monday. And in epistasis, what we're looking at is the idea that the one pair of genes can control the expression of another. And you can have a recessive type of epistasis where if you have a recessive pair of genes at one locus, that can overwhelm whatever's at the other locus. And so in that case, we had the examples of the mice, the albino mice, regardless of whether or not they were supposed to have a gouty or black pigmentation based on the A locus. If they were homozygous recessive at B, they became albino. In dominant epistasis, if they had the presence of a dominant allele at one location, in this case locus A, you had white fruit. So just that one dominant allele, capital A, was able to overwhelm everything else. So in a recessive epistasis, you see a handful more of the recessive phenotype. In a dominant epistasis, you see a lot more of the dominant phenotype. And then the third interaction that we left off discussing was when genes work together to make a phenotype. And in this case, we have a situation where you have to be at least heterozygous for both loci. If you're homozygous recessive at one loci or the other, you lose the phenotype. There's only two phenotypes possible, the dominant and the recessive. And so you have to have a dominant allele at each locus in order to get the dominant phenotype. Okay. So what we have in this case, this is the last example here of these kind of epistatic interactions, is a type of epistasis where you end up seeing a novel phenotype in the F2 generation. And so in this case, they are talking about squash. I love squash, so I think this is it's a good food to talk about. But squash apparently have weird inheritance patterns with squash shape. And so the dominant phenotype for a squash is for it to be disc-shaped, this particular summer squash. The recessive phenotype would be for the fruit to be long instead of disc-shaped. So when you take a true breeding disc-shaped squash, you cross it with a true breeding long squash, your F1, which are going to be your heterozygotes. So your big A, big B, little b, are all going to be disc-shaped. So because you have dominant alleles at both loci, you end up with this disc-shaped fruit. Now, what happens when you cross the heterozygotes, you cross that F1 generation. So remember, you'd have 3/4 of the offspring would carry the big A, 1/4 would carry the little a. 3/4 big B, 1/4 little b, 3/4 big B, 1/4 little b. So what happens in the F2 in this type of interaction is you end up with a novel phenotype that appears. And that phenotype appears anytime you have a mix of one locus having a dominant allele and the other locus being homozygous recessive. So here, in this case, big A, big B, you're going to have both alleles, both loci have a dominant allele. These fruit are going to look normal for that genotype. They are going to be disc-shaped fruit. Now here you have at least one dominant allele for the A locus, but these offspring are homozygous recessive for the B locus. And in this type of interaction, that generation of the novel phenotype, what's happening here is whenever one locus or the other is homozygous recessive, you end up with this weird new spherical shaped fruit. So then we can go back here and we see homozygous recessive at A, dominant, at least one dominant allele at B. So that's another 3/16. Again, homozygous recessive at one of the sets, at one of the loci. These are also spherical fruit. Now, when you have homozygous recessive at both loci, you end up with the homozygous recessive long fruit. So what it's trying to show here is that neither locus is more important for the other for shape. Both loci contribute, because if either locus is homozygous recessive, you end up with this weird phenotype. And technically, I guess if we looked at these squash, that spherical shape is almost an intermediate between the classic disc-shaped squash and the long squash. So here you've got this kind of middle guy that's sort of not long, but also still kind of wide like the disc. Yes?
So would it be that a third phenotype is introduced, or that it's a intermediate?
So in this case, it's kind of the same. It's kind of the same thing. Either it's not really an incomplete dominance because it is not based. Just incomplete dominance is really based on one locus. So if it was one locus that controlled shape, yes, we would call it incomplete dominance. But because we're looking at two loci and one has to be homozygous recessive, they call it this kind of complementary interaction, or this novel phenotype. The other way you would know that this is a classic incomplete dominance is if we saw the spherical fruit as the F1 generation. So that's a really good question. If we were looking at incomplete dominance, we would expect this individual to be spherical, but this individual is disc-shaped. It has the dominant phenotype. And so what we're really looking at here is, because both of these pairs of alleles are necessary for shape, if you've got dominant at both, you'll get the dominant phenotype. If you have recessive at both, you'll get the recessive phenotype. But it doesn't really do the right thing when you've got dominant at one and recessive at the other. And so they're calling this novel phenotype, and they're saying that these genes are both equally important because phenotypically also you cannot tell the difference between this squash and this squash. So even though by genotype this group of squash is recessive at the B locus and this group of squash is recessive at the A locus, phenotypically they're identical. Both genes are playing a role in shape. And so for the disc-shaped fruit, you have to have a dominant allele at both loci. For the long fruit, you have to be completely recessive. And then for that spherical fruit, that novel phenotype that shows up in the F2 only, you have to be homozygous recessive at one locus or the other. So these are some unique gene interactions that can influence phenotype. So this is why, again, it was really lucky that Mendel chose the traits he did, because, you know, if he started with a round seed and a wrinkled seed, and then somewhere in the F2 he popped up with a square seed, he wouldn't be able to, or he would not have been able to come up with the same theories that he was able to come up with. So other extensions of Mendelian genetics also involve the fact that not every single gene only controls one trait. There are many genes in all systems that actually can play a role in many, many phenotypes. And that phenomenon is called pleiotropy. We're looking at one gene that, when mutated, has multiple effects in phenotype. So they give an example here of a condition called Marfan syndrome. Marfan syndrome is inherited in an autosomal dominant fashion, meaning you only have to have one copy of the mutant allele in order to experience Marfan syndrome. The gene is called fibrillin. It's involved in forming things like connective tissue. And so what happens phenotypically in a patient that has Marfan syndrome is tied to this defect in connective tissue. So individuals with Marfan syndrome are usually very, very tall. They have really long extremities, very long fingers and toes. They normally have a slightly caved in chest. They have issues with the blood vessels in their heart. That's usually what leads to an early death in Marfan patients. They can have issues with their bones. They can have issues with their eyes. So this mutation in one protein in the human body causes effects systemically. So this is an example of pleiotropy. Abraham Lincoln was thought to have Marfan syndrome because he was so tall, and he was always he always had a lot of pain. So they've gone back and, posthumously diagnosed him with Marfan. If you ever have seen the show Rent, the guy who actually wrote Rent had Marfan syndrome, and that's why he died, because they usually have very weak hearts. So one gene, multiple phenotypic effects. And so far, we've only been talking about one gene, one effect, or maybe even two genes, one effect. So at this point, using this term pleiotropy, we can refer to a single gene that has multiple effects within the host. So any questions on pleiotropy? So sometimes they're these linchpin genes that are super important. And when you can't form a structure like your connective tissue, you're going to notice it all over the body. Okay, so we have spent the entire time talking about inheritance and talking about genetics, focused on the autosomes, or the non-sex chromosomes. But there are numerous, at least in organisms that have X chromosomes, there are numerous genes on the X chromosome, and they lead to very interesting inheritance patterns. So in plants, in animals, usually the sex of the individual is determined by a pair of chromosomes called the sex chromosomes. In Drosophila, which are the fruit flies, in mammals, we use the XY system. Birds use a ZW system. Reptiles have a really weird system of sex determination that involves not only chromosomes, but also temperature. So at least in Drosophila and humans, we have X chromosomes. And males are what we call the heterogametic sex, because the two chromosomes are not the same. In these individuals, these particular species, females are for the most part the homogametic sex, because the two sex chromosomes are the same. And in birds, it's opposite. In birds, females are the heterogametic sex and males are the homogametic sex. So sex determination is not as simple as X and Y. When we talk about, in this example, when we're talking about organisms that use the XY chromosome system, males are generally the heterogametic sex, The other term we use is hemizygous. And the reason for that is that for every gene on the X chromosome, males are neither homozygous nor heterozygous. They don't have a match. So in males, let's talk about human males. In human males, for the most part, human males have one X chromosome. They have one copy of every gene. Human females, for the most part, have two X chromosomes, two copies of every gene. So females can be homozygous or heterozygous for different alleles at each locus on the X chromosome. But because males only have one copy, they are hemizygous. And this is why, as we proceed through this section, what you'll see is that many times when a gene is on the X chromosome, if there is a mutation that leads to a phenotype, that phenotype is almost always seen in males. Because if males have a mutation on their X chromosome, they have no backup. Females who have a mutation on their X chromosome should have a backup. So that's why things like hemophilia, color blindness are all much more common in males than in females, because males inherit one bad copy from their mom, but females can maybe have a good copy to compensate. So again, hemizygous refers to the heterogametic sex. Whichever sex has two different sex chromosomes, you call them hemizygous because they usually don't have a match. And that's why you see a very unique inheritance pattern with these genes. And we call it X-linkage because these genes are on the X chromosome. And some of the first studies with X-linked inheritance were done in the 1920s in Drosophila. And there's a mutation in Drosophila that leads them to have white eyes. But what he saw was that for the most part, the sex that most commonly had white eyes was the male. And so that led him to determine that the locus encoding the gene that led to eye color is actually on the X chromosome. And so what they're showing here were his results. And the other reason that he came to the conclusion that this gene was on the X chromosome is because it also mattered which parent carried the white eye allele. And so we can do a Punnett square for X-linked traits as well. And so let's take a look and say we're looking at this example. And I'll show you how to do the Punnett square for something like this. So if we're saying that based on his experiments, we know that the gene encoding eye color in fruit flies is on the X chromosome. It's a sex-linked trait. And so we're going to cross a female with red eyes with a male with white eyes. And what we're going to assume in this case is that both of these are true breeding. So you have a female. So she has two X chromosomes, both carry the red allele. You have a male. He has XY. Because he has white eyes, you know that his X has the white allele. And in this case, red is dominant to white. So in a situation where a female fly might carry both the red and the white allele, she will show red eyes. So if we did this cross here, the male fruit fly is only capable of transmitting the white allele. and he only gives an X to his daughters. So every one of his daughters is going to inherit the white allele. All of his sons get a Y. Kind of weird to think about daughters and sons and fruit flies, but that's okay. If we consider that the female is a true breeding red eye, that means all of her offspring get an X that would encode a red eye. So the daughters that result from this pairing are heterozygotes. They have the red and the white allele, red and the white allele. The sons that result from this pairing are going to have the normal phenotype because they have one copy of the X chromosome and it is normal. Now, if we look at this, the idea would be you have 50% female and they all have red eyes. 50% of the offspring would be male and they all have red eyes. Then you cross them. So you cross the carrier female with the normal male, because that's what resulted. Okay, and what you should get out of a cross like this is that 50% of the offspring will be female. One is going to be homozygous dominant for red, one is heterozygous. But either way, all of those females have red eyes. You also get 50% of your offspring are male. However, we now have one male that has the red allele and one male that has a copy of the X chromosome that would encode white. So in this case, you would have 25% of your offspring would be males with red eyes, 25% of your offspring would be males with white eyes. So when you start to look at pedigrees for traits like this, that are recessive alleles located on the X chromosome, you generally see those traits in males. It's just that much more likely. Now, on the other side, he did manage to make females with white eyes. That would be easy enough. You would just continue to cross carrier females with white-eyed males. Because as soon as you do this, at least half of your females are going to have white eyes. So it can happen. It's just much less likely. So if we look at the other example of a female with white eyes and a male with red, what you end up seeing in this case is all of the female offspring are very lucky because dad had a normal X chromosome. And so because dad had the normal X chromosome, even though both of moms were defective, the female offspring got the normal one from dad. They all have red eyes. Unfortunately for the males resulting from this cross, because both of the females' X's are recessive, all of the males have white eyes. And so in this case, you see a ratio of 50% female offspring with red eyes, 50% male offspring with white eyes. And then you can do the cross again. So we have carrier females, and we have white-eyed males. And in this case, you now have 25% of your offspring are females with red eyes, 25% of your offspring are female with white eyes, 25% male, red, and then of course that final 25% male, white. And so the idea is that yes, it is entirely possible to get females, even though they have two X chromosomes, you can get females that express X-linked traits. But the only way that works is if a carrier female or a female who already has the disease reproduces with a male who also has the disease. That's the only way that you can get female offspring that are colorblind or hemophiliacs. And what happens with recessive conditions like this and others is that it's much more likely, let's say, if grandpa's a hemophiliac, all of the daughters are going to be carriers. And when you start marrying your cousins and you have a huge potential of lots and lots of female carriers reproducing with their male relatives, these show up much more frequently in the population. And so you can look at things like the royal family's pedigree for hemophilia, and you can see where they married their cousins and why it turned out that way. Okay. So what I'd like to do now for the last few minutes is we'll do a little quiz. So go ahead. If I did them all on Fridays, they wouldn't be pop quizzes. You would know they were coming. I like to have a little fun in my life. Quiz on a Wednesday. Yeah, my life. That's the one. Quiz on a Wednesday. Last name, first initial, please. Oh, see, look, it shouldn't make you feel too bad. It has a little heart, so it's happy. It's a happy, loving quiz. That love hurts. All right, and remember the pin will be at the bottom if you haven't been able to quite log in yet, so you can continue to log in. And so, oh, wait. Oh yeah, perfect. So with incomplete dominance, a likely phenotypic ratio resulting from a true breeding monohybrid cross is what? So do a Punnett square of a classic monohybrid cross, right? You're crossing homozygous dominant, homozygous recessive. What phenotypic ratio do you see in the offspring? So I'm sorry, you're crossing true breeding monohybrid cross. Your snapdragons.
Oh, the snapdragon. Oh,
What's your phenotypic ratio going to be? Draw your Punnett square if you have to. You've got 10 seconds left. Good. Because you're going to have one homozygous dominant individual, right? And you're going to start to see the heterozygotes, and they'll be pink. All right. With respect to genes on the X chromosome, mammalian males are homozygous, heterozygous, hemizygous, or complementary. Okay, about 10 seconds left. Good. Hemizygous, right? They don't have another chromosome to have a matched set of loci. They are hemizygous. The ABO blood group in humans is an example of what? Codominance, incomplete dominance, dominant epistasis, or recessive epistasis? Forget O. What does epistasis mean? No, I don't know if you did. It's codominance. Remember, epistasis involves two sets of loci. So if we're talking about one locus, you cannot have epistatic interactions. And because you have A and B that can be together, not a blended phenotype, it's codominant instead of incomplete. One gene having multiple effects on the phenotype is known as codominance, incomplete dominance, epistasis, or pleiotropy. Good. Pleiotropy. Remember, epistasis is two genes having an effect on one phenotype. Pleiotropy is one gene having an effect on multiple phenotypes. Blank kind of alleles encode proteins made with normal amounts with normal function. Loss of function, gain of function, mutant or wild type. So normal amount of protein, normal function of protein. Wild type. So remember, loss of function is either less protein or dysfunctional protein. Gain of function is better. Mutant could be loss or gain. Wild type is normal.